Well, hey now, living environment fans from across the globe, worldwide. It's time for another Bellringer. It's Monday. Hope you enjoyed the weekend. I know I did. And I hope you got out and enjoyed some of the amazing weather we finally had this winter. The sun hasn't been out very long, and it was really nice, especially Sunday. Wow, what a day. All right, so we're going to do the Bellringer. I don't know if you were like me. You spent most of the weekend thinking about DNA replication.
All right.
Hey now.
Mr. Kovacs class. It's Mr. Kovacs class.
You might learn a ton.
It's Mr. Kovacs class.
It's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class.
Our good friend DNA, right? You recognize the double helix shape, the spiral staircase there of the DNA molecule, the twisted ladder, if you will. And by now you remember the four chemicals that are the rungs, the four bases, the four nucleotides that are the rungs of the ladder. What are they again? Good, that's adenine, thymine, guanine, and cytosine. Those are the four, right, the nucleotides that make up the DNA molecule. The nucleotide is actually the whole thing. It's the sugar, ribose, plus the phosphate group, plus the base pair, like in this case A, adenine, right? And then they line up. A always goes with, very good, and G always goes with, very good, and they stick together by what are called hydrogen bonds, which hydrogen bonds are one of the weaker bonds that exist in chemistry, right? They're not super strong at holding this together. They're easy to break, which is good, because in order to replicate your DNA, you have to unzip or you have to break those hydrogen bonds. Replication is semi-conservative, because when you break the hydrogen bonds and then attach A's to T's, G's with C's, you get two brand new strands of DNA. Each new strand has one old strand. Each new double helix has one half of it is an old strand of the old double helix. So when mutations occur, they happen usually in the new strand copy. I introduced you on Friday is called helicase. Helicase is the enzyme that unwinds a double helix, right? It breaks the hydrogen bonds, unzipping the DNA molecule, right? Helicase. There's helicase, unzipping the DNA molecule. And then some other enzymes go to work. We call those polymerases. DNA polymerase. It goes around and speeds up the reaction of linking A's to T's, G's with C's. It's an enzyme, polymerase. Helicase is an enzyme, right? They speed up chemical reactions. What's one thing that might denature an enzyme? Good, yeah. Heat. Heat is really bad for enzymes. We talked about that. We did a lab where we studied that, in fact. All right, so polymerase makes the new strands by matching up A's with T's, G's with C's. It's interesting because polymerase has to go in two directions. Like there's one polymerase that goes this way, and then the other one has to go kind of backwards, which is kind of interesting. But they both do their work to make copies, and then you get new DNA strands, right? Talking about the life cycle of cells, right? Replication always occurs in what's called the S phase, the synthesis phase. This is where DNA replication happens. But cells spend most of their life in what's called the G1 phase. G1 stands for growth and development and living their life, making proteins. And then they have to get a signal. Something has to tell them, okay, it's time to replicate your DNA. Right? And then they copy all their DNA, they continue to grow, and then they split into two new cells. And so that's where we're headed next is, how does this whole mechanism work? The cells copying themselves. We call it the cell cycle.